Hello students, and welcome to the audio presentation focusing on chloride and its effects in the CNS as well as in the skeletal and smooth muscle. In this presentation we will be looking at gamma-aminobutyric acid, or GABA as it's also known as, benzodiazepine and its effect on neural excitability, the role chloride ions play in the skeletal muscle, the channels that are responsible for chloride movement in skeletal muscle, and the physiological function it has, as well as what channelopathy is and how it affects the conduction of chloride within the skeletal muscle. So GABA is a chemical receptor that functions as an ion chemical receptor as well as a G-protein coupled receptor, making it also known as an amino acid neurotransmitter, but for this presentation we will just be looking at its function as an ion chemical receptor. So GABA is an inhibitory neurotransmitter, meaning that hyperpolarizes its target cells by opening chloride channels and allowing chloride to enter the cell, making it less likely to fire an action potential. The local circuit transmissions of GABA allow it to alter the excitability of primary output neurons. So GABA is able to exert its physiological actions with its two classes of receptors, GABA A and GABA B. GABA A sites are known as ionotropic and are the major site of action. When GABA binds to GABA A receptors, it promotes a direct opening of a selective ion channel that's permeable to the negatively charged ion chloride. When chloride ions flow into the neuron, they hyperpolarize the membrane of a neuron, making them less likely to fire an action potential. GABA B receptors are metabotropic receptors, causing the frequent opening of potassium channels, allowing potassium to flow out of the neuron, making it hyperpolarized. GABA is able to move via GABA transporters, which move it from the synaptic cleft into neurons. GABA activates chloride channels depending on the dosage needed. The opening of these chloride channels requires the interactions of the agonist, diazepam, and is able to activate physiological responses over low micromolar concentrations of 100, as well as concentration-dependent desensitization, meaning that in the presence of continued GABA administration, chloride currents are not maintained but gradually decrease, and this occurs over seconds. Now, Valium is a benzodiazepine prescribed to patients with anxiety and panic disorders, as well as OCD, and ultimately reduces neuron excitability. When taken with depressants such as alcohol and painkillers, it will more than likely cause an overdose. This is because increased GABA activity caused by benzodiazepines will create a sedative effect. When benzodiazepines bind to the GABA receptors, they influence where the GABA will initially bind. This is an allosteric effect, increasing the effect that GABA binding has on the receptor, causing an opening in the ion channels, allowing negatively charged chloride ions to enter the channel, pushing the action of these receptors to polarizing the membrane potential, and if you can't remember what that means, it's less likely to cause an action potential. This in turn stops the neurons that make up networks which cause anxiety and panic. With the introduction of alcohol with Valium, a person would be increasing the amount of GABA receptors into the membrane channels, causing further depolarization. Now, channelopathies are conditions that affect ion channel functions, and your chloride ions are located in the plasma membrane and are able to actively transport chloride throughout the skeletal muscle. These are important as they play roles in cell volume regulation, transepithelial transport, secretion of fluid from the secretory glands, and the stabilization of resting membrane potential. There are 4 types of these channels, being the transmitter-gated chloride channels located in the inhibitory synapses, the ATP binding cassette channels, the voltage-gated anion selective channels, which is in the ceruloplasmic membrane of the skeletal muscle, and voltage-gated chloride channels, which are in excitable and epithelial cells. In regards to the voltage-gated chloride channels, CLC1 is the most important in skeletal muscle as it causes tetanic muscle excitation, as it contains a high amount of chloride, leading to a fast repolarization of the tetric tubal membranes, which then become depolarized by potassium. So a channelopathy is an inherited disease which ultimately causes a mutation within the proteins in an ion channel. A channelopathy that affects chloride is cystic fibrosis. This is because as the CLC1 channel is mutated, the nerve and muscle are unable to be stimulated the correct way, causing the smooth muscle, especially in the lungs, to not be able to contract properly, and therefore inhibiting the central movement from the muscle occurring.